Hi, this is Miss Litton, and this is Lunchtime with Biologists. Say hi.
Hi.
All right, so we're still continuing with big idea number 1 as we're reviewing for the AP exam, and now we are at Enduring Understanding 1B. Organisms are linked by lines of descent from common ancestry. Some of the images here are going to be similar to some of the ones we saw in Enduring Understanding 1A. So sometimes they come up just because they're applicable to both. So I just want to tell you about that. And then I'll find it. Here it is. Okay. So we know here, this is showing you some kingdoms, but we know the domains. What are the domains? Archaea, eukaryotic, bacteria. So I would have a good description of each of those and then Venn diagram it. How are they similar? How are they different? That would be a good way to review it. And then within domain eukarya, you have the kingdoms. What are the kingdoms underneath the domain eukarya? Animalia. I heard somebody say mammalia. That would be, no. It's class Mammalia under subphylum Vertebrata, so no. Okay, but you know that it's Animalia, Plantae, Fungi, and Protista. Those are your 4 kingdoms underneath domain eukarya. Again, I would have a running description of each of those 4 kingdoms. You have summary tables in your notes and in your textbook and pretty much anywhere online. If you have a representative animal and key characteristics like autotrophs or ingestive heterotrophs or absorptive heterotrophs. Okay? And then we looked at the characteristics of life, which all living things have. So let's say that together. Living things are organized, that's made of cells. Okay, let's do it together. They acquire materials and energy. They homeostasis. They reproduce. They grow and develop, and they adapt and die. So all living things have those in common. Are viruses alive? No. So they don't meet those characteristics. So they are not. Okay, so we looked at what are some similarities. The instructions for life. That's a conserved process. DNA, ATP. We looked at this before, right? We looked at the processes of cellular respiration. We looked at similar proteins. So one starting point here is the DNA. Another one is the genetic code. That is universal. That's similar. Why don't we have, this is a really good review. Why don't you have the person sitting in the blue seat? Why don't you explain this diagram to the person sitting in the slated seat? Go ahead. Don't write on this. Write on this. Name, period, and version up at the top. Thank you. Okay. Are you good on this? I just want to ask you a couple of things. You see where it says gene 1, gene 2, and gene 3? Would those be introns or exons? Exons. They will be expressed, right? Remember that? Remember how we said cut out the introns before it leaves the nucleus, and the exons get expressed. Now, you could have genetic recombination in which of those genes are actually remain, because some of those genes could be spliced out. Where do we see variations in that? Immunity, sure. In immunity and all the millions of variety we have there, that would be a good example. Okay, so we see the DNA template, and why are they showing you 3' to 5'? What is that referring to? Yeah, they are anti-parallel. One strand runs from 3 to 5, the other one runs from 5 to 3, but you're counting the carbons, right, in sugar. Remember how 1 is the one that's connected to the nitrogenous base. Remember how we went around the carbons, and this is the template strand because DNA and RNA polymerase read from 3 to 5, making complementary 5 to 3. Okay, so here you can see the RNA. What does the M stand for? Messenger RNA. Now, they're just showing you one RNA strand there, but it could be, you have to look at it, is it before or after it's been modified? Do you remember the ways we protect it?
Cap with the
Cap, 7-methylguanosine, and a tail, 100 to 200 adenines, and then you cut out the introns, right? Leaving the exons, and that would be the mature RNA that's going to be translated. Every 3 bases on the mRNA is called a codon. It's complementary to a anticodon located on the tRNA. Okay, so that's what you're seeing. Each tRNA would bring 1 amino acid. This is transcription is going from DNA to RNA. Using the RNA to build a protein is called translation. Translation occurs where? Where's the workbench for that? Ribosomes. Okay? So if what I just said right there was a little bit shaky for you, you want to go back and review transcription and translation, those terms. All right, so this is mitochondrial DNA. That can be used for comparisons, especially, would this be prokaryotes or eukaryotes? Prokaryotes don't have mitochondrial DNA because they don't have mitochondria, because they don't have any organelles, right? So you would need a eukaryotic cell in order to have mitochondrial DNA. Does that make sense to you? Think it through? Okay, go ahead. What were you going to say?
Like, we should argue that since mitochondria were prokaryotes, we compare prokaryotes to mitochondria?
Well, if you say you have mitochondrial DNA, you're in a eukaryote. Yeah. All right. So you could look at mitochondrial DNA and use that for comparisons. Mitochondrial DNA is only passed through the female because remember the male, Mr. Happy Sperm, when he fertilizes the egg, he's just bringing the DNA. He's not bringing anything else. So you could trace in each of your cells right now, your mitochondrial DNA will be the same as your mother, which will be the same as her mother, which will be the same as her mother, and so on and so on. I don't remember what this full tab is. Oh, okay. So here you can just see the different processes, DNA, mRNA, making your protein, and the similar process occurring in the mitochondria. If you remember, let's talk about what you were referencing, the endosymbiotic hypothesis, which is how eukaryotes could have evolved from prokaryotes. Is that sounding familiar? And what he was referencing is that that mitochondria and chloroplasts could have been free-living prokaryotes that went to live inside of another prokaryote. And what is our evidence? Do you remember some of the pieces of evidence for that? Mitochondria and chloroplasts, what? Have their own DNA. They have a double membrane, which they could have picked up the second membrane when they entered the cell. They're in, what else? What? Oh, yeah, that would be evidence. Yes, I agree. What else about mitochondria and chloroplasts? They're about the same size as prokaryotes. They have the same ribosomes as prokaryotes. They can make proteins as well inside. They are self-replicating. Okay, those would all be pieces of evidence for that. So you just want to remember that you have DNA inside the nucleus and also inside your mitochondria and potentially your chloroplast. So who does glycolysis? All cells. So that would be another, conserved process because all organisms do glycolysis. Is it an aerobic or anaerobic activity? Anaerobic. Does not require any oxygen. What do you start out with? Glucose. How many carbons? 6. What are you going to do? Break it in half. How many steps?
10.
How many enzymes? What are these called? 2 pyruvic acids. And you reduce some NAD and make some ATP. 2. Now, did we really only make 2? No, we made 4, but we need 2 ATP to get the party started. Good. All right. Keep going. Here, prokaryotic versus eukaryotics. Last time, who went? Blue? Slate, why don't you differentiate between prokaryotic and eukaryotic cells? Go ahead. Oh, can you write the verse? Oh, you did. Yeah, you want to write, I'll do it for you. Chapter 37. Oh, sorry. Thank you. Okay, now, anything terrifying so far? The whole thing? No, not tissue. Okay, not everything is terrifying, right? There are some things maybe you want to revisit, but hopefully it's sounding familiar. So this is just a little diagram. You could find diagrams too. It would be very easy just breaking down the 3 domains, bacteria, archaea, and eukarya. Which one are the extremophiles? Yeah, archaea, because they live in these, like, some are methanogens, halophiles, thermoacidophiles living in these extreme environments. Okay, something else we look at is phylogeny. What would this be? Like a cladogram, exactly. You keep dropping off characteristics. We talk about shared derived characteristics. What do they have in common? What traits do they share? So this one is a common ancestor to all of them, so it must have traits, right, that all of them have. But then we're branching off here. B and C have some traits that A doesn't have. That's why we've got a branch. Okay? So when you look at a phylogenetic tree, when you construct it, the systematics, Blue, why don't you say what systematics is? Go ahead, Blue. And Slate, this will be you. So systematics is finding and studying all the different things that you can use to construct the phylogeny. Okay, so when we construct that phylogenetic tree, you can see it goes from cellular to population level. So you can look at characteristics of the cell, right? You can look at the DNA, you can look at the proteins, you can look at the anatomy, okay, in order to come up with your phylogenetic trees. Okay, so a taxon is the name applied to a taxonomic group in a formal system of nomenclature. We talked about cladograms. It says a clad down there. Blue, go ahead. Tell what a clad is. This should be familiar. Okay, now, these two right here are, this is a clad, but this is not a clad, and this is not a clad. Explain, whoever's turn it is, I don't know who it is, why are the two on the bottom not a clad? So the one on the right is not a clad because they're not including a group, right? And this one is not a clad because they're going over and getting one from another line. Okay, you could call the whole thing, you could call that a clad, but we can't take a little section, take two of these and one of these. Now, those are just sticks and lines on a page. But what that's saying is you wouldn't look as a trait as being differentiating if these are the only three that shared it. You understand what I'm saying? If these are the only three that have that, then you're not going to pick that out. Because this should be a trait that is shared by all within that line of descent. Okay, and so this is a good one where it keeps saying from here on up we have jaws, from here on up we have lungs, from here on up we have claws. Birds are a problem, right? Yeah, birds are a problem the way we classify them. They maybe should be within where should we put them possibly? With what? Reptiles, exactly. All right, and this is showing you how you could list off all the traits. I wouldn't be surprised if they would have you make a cladogram for your AP exam. and they would give you a series of traits, and then you would say, okay, what do they all have? That's good trait's going to go at the bottom. Then somebody's missing it. Dump him. Keep going with everybody else. Dump him. Keep going with everybody else. Okay? And if you had to make a cladogram on your AP exam. Okay? If you take a look, if you look at this trait, what you want, are you going to make up your quiz? Yes, no problem. What is parsimony? What does that mean? Do you remember? Did we talk about that? When you talk about parsimony, you're looking for the least amount of unexplained events. And what you're trying to do is you're trying to don't have something evolve twice. Have a common ancestor. Don't say that it evolved here and then this trait evolved over here. Just have it evolve one time. Okay? One time for all. If you keep having something pop up here and pop up here, pop up here, that's not going to be a good organizational strategy. All right, and okay, I think we went over that. All right, let's take a look at this. This is some other characteristics we could look at. Remember, let's go back. I just want to make sure you know where you're at. We're looking at organisms are linked by lines of descent from common ancestry. This is all that we're supporting, the similarity. Okay, so here, where are you? We looked at homologous structures. What do homologous, why doesn't it blue tell slate? What are homologous structures? Put it in your own words. All right. So homo means same, and these are homologous structures, so they have the same anatomy, which means they probably have a what? Common ancestor. Because what's going to code for that anatomy? What determines the DNA? They may use it for different functions, but because they have the same bone anatomy, they probably had a common ancestor. What wouldn't we use as a piece of evidence? We wouldn't use analogous, because that just means same what? Function. Which means they're responding to their environment in the same way. It could be flying. But a butterfly is not closely related to a bat, right? Because they don't have the same anatomy to fly. All right? Let's take a look at this. We have 3 different types of evolution right here. And why don't, whose turn is it? Slate? Is it Slate? Mm-hmm. Do Divergent. Go ahead. Okay, so when we look at Divergent Evolution, they had a common ancestor somewhere. Like if you look at seagulls and penguins, do you think they're related? Seagulls and penguins. Yeah, why? They're both birds. They both have wings. Do they use their wings for the same purpose? No. What does the penguin do? He swims with them, right? And the seagull is going to do what? Fly. But if we looked at their heart structure, if we looked at their anatomy, if we looked at their lungs and their parabronchi, then and their anterior and posterior air sacs, we might go, oh my gosh, they are related. They just use, and we could look at their homologous structures that I just named. We could look at similar anatomy and go, they must have a common ancestor. They just are exploiting their environments differently, okay? As opposed to, I don't know what the next one I have here. Talk about convergent evolution. Other bio buddy, go. Try to put it in your own words. Own it. So in this way, we look like the example here is fish and dolphins both have fins, but are fish and dolphins closely related? No, okay? They are not, and they don't have a common ancestor, they just have structures that are similar, okay? Parallel evolution is like, we have a common ancestor and we have similar structures. Okay, and that right here would be a good example. When you look at placental and marsupial mice, they had parallel evolution. They are mammals, they have a common ancestor, and they have similar structures right now. Of those three lines, okay, divergent, parallel, and convergent evolution, those are all, when you look at traits and when you're making and generating a phylogenetic tree, you need to look to see if any of the, what kind of evolution is evidenced by that. All right, next. This is showing you classification, and I think we learned them, right? Let's say them together. Domain, then what? Kingdom, phylum, class, order, family, genus, species. What is the most inclusive classification? Most inclusive? Domain. The most exclusive? Species. Good. Right? I don't know how many questions are here, but you can go ahead and log in. However you would like to log in. Oh, wait. I'm going to remove because there's some on here. Go ahead, log in. Whatever you want.
Oh, I need to put in the code. You should put the code in this side too.
Just me.
Your name.
Just do that.
Yeah.
No, smoke no.
Guys, are you remembering you have half days tomorrow?
Yeah.
You're going to have to move fast through the curriculum tomorrow. Speedy. Ready? No, you're taking this early, so you don't have to think about it. Right on here, not on here. And then, your bio. You get stuck on a couple.
Really?
Yep.
Okay. Thank you.
You ready? Okay, which of these are not eukaryotes? So what did you mark? Bacteria and Archaea? That's true. Eukarya is a eukaryote. Animal, plants, and fungi, those are all what? Kingdoms within eukarya. Okay. So 6 of you struggled. You put A, B, and F. F is a kingdom underneath eukarya, fungi. Okay. Which kingdom is mislabeled? Fungi, prokaryotic, single cells, because fungi are eukaryotes, not prokaryotes. Plantae are multicellular. D, Animalia do include arthropods and humans, and Protista are unicellular eukaryotes. So you might want to review just those kingdoms, just to have a good overview. Which of these characteristics is shared by bacteria and Archaea? Wow, 2 of you got that right. Yeah, okay, so let's go over it. Good, nobody picked A. That is hashtag awesome. Okay, they do have absence of a nucleus. Did everybody get B? No. Okay, they do have ribosomes. Absence of membrane-bound organelles. That is true because bacteria and Archaea do not have any organelles, and they do have a cell wall. Okay, so that one maybe review a little bit more again on your 3 domains and your 4 kingdoms. I would review that based on the 2 of you getting that right. All right, and last but not least, when you look at bacteria and their ability to get new DNA, there are 3 different ways here. Could you differentiate between those 3 different ways? Why you might have evolution in bacteria, how they could have new DNA? Whose turn is it? Okay, Blue, go.
I would talk to you about
Okay, I'm going to just jump in here so we can finish. Transformation is when you get new DNA, maybe from what? From your environment, from other dead bacteria. Conjugation is when 2 bacteria have sex with using their sex pellets. Transduction, virus acts as a vector to take DNA from one bacterium into another. Perfect. You did. One more enduring understanding. How many do we have left? 53 left. Awesome. All right.